everyone, this is our pre-lab recording for Lab 1.3, our first face-to-face lab. So just to remind you where we are in our Module 1 of Plant Biotechnology, this is the week that we're going to get to use our pipetting skills and load samples into an agarose gel and run that agarose gel and see the results. Okay, so our plan is that, of course, we'll have our pre-lab quiz 1.3. This is due via Blackboard at 1:30 p.m. Monday, just like usual, for everybody. We're going to have an overview of pipetting and agarose gel electrophoresis in this recorded pre-lab lecture. And this is going to introduce the concepts and techniques that you need to know for the in-person lab and for the worksheet. You need to plan to attend lab or let me know ahead of time. You will also have time to work on your draft version of your figure and legend. The worksheet is going to be due Tuesday, September 21st at 11:59 p.m., so you'll have plenty of time to get that done. It's rather short, just 10 points. And that draft version of your figure and legend from Lab 1.2 is not due until September 27th, but the upload link is available now if you want to get that done. So since this is our first face-to-face lab of the semester, we need to review some lab safety. Masks are required. Backpacks, jackets, and phones should be stowed at the front table area to get them out of the way. There's no food or drink in the lab. And everyone should wear long pants and closed-toed shoes for safety. And lastly, you should wear gloves when you're told they're necessary, and gloves are necessary when we're loading and running an agarose gel. So, in terms of when to come to lab, we're going to follow the plan that was on the last slide of the Lab 1.2 PowerPoint notes. I've also added that slide here in this lab set. So that's going to tell you if you are supposed to attend lab at 2 p.m. or if you are supposed to come at 3:30 p.m. When you get to the lab, you'll get to work right away. You'll use a micropipetter to load different DNA molecular weight standards into an agarose gel, and then you'll resolve those fragments using gel electrophoresis. After about 30 minutes, you'll be able to view the results of the agarose gel on a transilluminator. And while the gel is running, you'll pour a new gel for the next lab to use. After lab ends, or before lab, at any point in time, you can meet virtually as an option with your lab group to complete worksheet 1.3. So again, it's up to you if you want to try to make a plan to do that with your lab group or just do it on your own and submit that on Blackboard, again, Tuesday by 11:59 p.m. And then just as a reminder also about the figure and figure legend. So this is just a reminder of who has been assigned to either 2 p.m. or 3:30 p.m., and this is pending online requests. If you do not attend lab in person for any reason, that's not a problem at all. Please, again, just let me know at least 24 hours before lab so we can make plans accordingly. Again, whether you're attending lab face-to-face or not, you can meet virtually with your lab group to work on worksheet 1.3. Okay, so what do we need to know for Lab 1.3? Well, you need a refresher on micropipetters. So in Lab 1.3 Part 1, there's information about plasticware and a pipetting exercise. And I know that a lot of you have learned about using micropipetters from BIO211. So you want to make sure you're paying attention to the limits and ranges of micropipetters. For P2, it's the smallest range of volumes. P20 is a little bit more. P200, the volume is even greater, and P1000 is the largest of the volume ranges, 200 to 1000 microliters. There's also a review of metrics in the lab manual, so you want to make sure you understand that and how to convert microliters to milliliters. There's a little table to give you some practice. If you get stuck, please feel free to email and ask questions. That will be helpful to know before the quiz. And there's also a micropipetter demonstration video with the link on the next slide. And if you watch that at the 2-minute mark till the end, so it's only about 2 minutes total, it'll be a really helpful refresher on how to use a micropipetter the correct way. Okay, and there's our little images of those different pipetters. So that's reminding us that there's 3 positions of the plunger, the rest position, the first stop, and then that second stop. And hopefully that looks familiar. But the video, again, will help a lot. So there is the link, so please watch that before you come to the lab. And then just a couple of tips and tricks to mention. So we're not going to be completing the exercises in parts D1 to D3 of the lab manual. And again, this is partly because we know you've pipetted already in BIO112. But we do want to make sure that you still understand how you would select the correct pipetter for a particular volume and how to determine the volume a given pipetter is going to deliver if it's dialed in at a particular setting. Some common mistakes to avoid. It would be, not using the correct plastic tip. So the different tips, they're usually yellow and blue, sometimes clear. They're different sizes. So different tips go with different pipetters, and that's important. And we don't want to break the pipetters. They're actually pretty expensive. So when you're drawing liquid into the plastic tip, you need to really release on that plunger slowly and not allow liquid to be drawn up into the barrel of the pipetter because that ruins the gasket. And then another good tip is that once you've drawn liquid into the pipette tip, you want to actually look at it and make sure you can see that liquid before you transfer that volume to the delivery tube. Okay, so in this lab, we're going to be using our pipetting skills to load an agarose gel. And the specific tip here is that we want to avoid puncturing the bottom of the agarose gel with a pipette tip. So the agarose gel, it really has a consistency similar to Jell-O, so it's pretty fragile. And those plastic pipette tips can be rather sharp, actually. So this cartoon shows you very nicely that you should only insert the pipette tip about halfway down into the gel's well. You do not want the tip to touch the bottom. That will easily make a hole and make it impossible for your DNA sample to run correctly. Another really important tip is the second one that says gently push on the plunger to release the DNA into the well. So you just want to do this very slowly with your thumb, and then keep the plunger pushed down all the way until you take the entire tip out of the well. And I'll remind you of that tip while we're in lab. So this diagram shows us the steps of loading an agarose gel. So step 1 is just showing you there are 3 wells. Step 2 is the pipetter is actually starting to load the sample into the first well, and then you would proceed to loading the second and then the third wells. And then at step 5, it's showing you what those samples are going to do as they move from the negative end to the positive end. So after you've loaded your samples, the ions in the running buffer are going to conduct the electrical current to move the negatively charged DNA towards this positive pole, which is at the bottom. And then one other note is that the molecular weight marker is also called a DNA ladder, or a molecular weight standard, or a ruler. So it has a lot of different names. But whatever name it's called, it's really important because it allows you to determine the size of your DNA fragment. So there are some other safety rules with electrophoresis. You want to make sure you turn off the power before you are manipulating anything in the gel box. You don't want to touch the gel or the buffer if the power is on. You want to make sure your gel is solid before you load and run it. You want to also make sure that you write down what's the order of the samples you've loaded. That's going to be hard to remember and important when you want to try to interpret the results. And as I've mentioned, we always wear gloves when we are touching buffer or the gel. You also want to put the leads on correctly. So what I mean by this is keeping in mind that black is the cathode, red is the anode, and you want to attach the leads red to red and black to black. So the power supply, which is this little guy here at the bottom, also has these colors, black and red. And so you want to just make sure that you get the correct colored lead into the correct colored hole. Okay. Otherwise, you could actually retrophorese your sample and the samples will run backwards, go right off the gel. Okay, so that's pretty much what you need to know about micropipetting and gel electrophoresis for Lab 1.3. As a reminder, what we're also working on is this draft version of a figure and a legend. So this is just a reminder of what we talked about last week. And so you are going to want to crop and annotate the gel picture that was provided in Blackboard. And remember all of the tips we talked about for writing a good figure legend. Okay, so that's just a reminder about that. And when you're ready to upload that draft, you're going to do it in the Peer Editing tab, which is currently in our Lab Blackboard shell. But remember, it's not due until right before Lab 1.4, September 27th. There are special instructions for uploading the draft in Blackboard Peer Editing. And then looking ahead to next week, make sure everyone prepares for the Pre-Lab Quiz 1.4. That's going to be based on parts 1 and 2 of the lab manual, Lab 1.4, parts 1 and 2. We're going to talk about DNA sequencing and actually do the analysis of the GAPC gene sequence. And we're going to find out how similar is this kale GAPC gene that we've amplified, how similar is it to cabbage and Arabidopsis DNA. You also need to read pages 21 to 26 in the Macmillan Writing Manual. So the quiz will be based also on this information in the writing manual, specifically in the sections called Reading Scientific Papers and Avoiding Plagiarism and Taking Notes. So make sure you look for those two topic headings, and sometimes those page numbers differ in different editions or in the ebook. Everyone has access to the ebook with inclusive access. So that's in our Lab Blackboard shell, in the writing manual tab. And again, just a reminder that some of the quiz 1.4 questions are from the Macmillan Writing Manual. And lastly, just make sure to complete that quiz at the usual time, Monday at 1:30, 30 minutes before our lab. So I hope everyone's looking forward to the first face-to-face lab, and I will see you there.